Hello everyone. In this presentation, Neuroproof would like to introduce you to our amyotrophic lateral sclerosis assays with motor neuron cultures derived from human-induced pluripotent stem cells. Amyotrophic lateral sclerosis is a severe disease with only partially understood etiology. Human iPSC cell-derived spinal motor neurons open the door to establishing innovative and predictive screening assays for developing new therapies for ALS. Clinical and in vivo observations support the hypothesis that a hyperexcited state of neuronal microcircuitry causes ALS by excitotoxic impairment of motor neurons. Neuroproof offers screening services of such disease models based on functional screening with multi-electrode arrays. Let me illustrate the multi-electrode array recording system first. We use cryopreserved postmitotic motor neurons in suspension and cultivate them on MEA plates with 48 cells and 16 electrodes. The motor neurons form stable and electrical active networks for several weeks. We then record the electrical activity with a Maestro recording station from Axion Biosystems in Atlanta, USA. With the MEA system, we record the extracellular action potentials of single neurons in a complex network. Here, you can see the recorded voltage train from one electrode representing the electrical activity of one neuron. Each timestamp of such an action potential is called a spike. With a spike detection algorithm, we calculate from the voltage train the so-called spike train from a multitude of neurons. When you record the spike train from frontal cortex neuron, you get a spike train as seen in image A. When you then apply to this network a compound like diazepam in a concentration of 300 nanomolar, you will see an activity as seen in image B. It is also possible to apply another compound, such as picrotoxin, to the native network. The exciting impact on the neural network then results in a spike train as seen in image C. Neuroproof has developed a specialized software to analyze these spike trains. We can calculate spike train parameters that describe the spiking activity of a spontaneous firing neuronal network. We divide these parameters into 4 categories: general activity, burst structure, oscillation, and synchronization. One general activity parameter is the spike train, reported as the number of spikes in a given time frame. Bursts are short episodes of high-firing activity. The number of bursts in a given time structures, which we call the burst structure. For example, in graph A, we see bursts with different burst structures. Oscillation parameters measure the regularity of periodic events, as seen in image B. Our last parameter describes the synchronicity of spiking activity. The synchronicity of spike trains is a sign of the synaptic connectivity of such neural networks. For our assays, we use both diseased and healthy motor neurons. Motor neurons are the first vulnerated neurons in ALS. Motor neurons are found in the spinal cord, the brainstem, and the motor cortex. Affected neurons from these different regions cause different symptoms of ALS. It is possible to differentiate spinal motor neurons from human iPSC in high quality and purity. There are known sporadic and familial forms of ALS. However, familial forms only have a minor prevalence of about 10%. In our assays, we use cell material from patients with familial forms. One cell line has the SOD1 mutation, and the other the C9ORF72 mutation. It is supposed that both mutations have a different etiology in ALS. Our assays are built with motor neurons received from brain cell in Madison, USA. In culture on MEA plates, these cells show first activity after 7 days in vitro, which is growing over time. Multiple comparisons showed sufficient reproducibility. After 3 weeks, we lose the clear bursting structure. With immunostaining, we see MNX1 and FOXP1 as markers of motor neurons and especially of spinal motor neurons. We also observe an increased number of electrical active electrodes, with a maximum number reached after 11 to 15 days in vitro. This time window determines the time point of the final readout of our ALS assays. Now we would like to discuss the functional phenotypes of these motor neuron cell lines. We can see after 11 days in vitro clear different activity patterns. Let us first look at a gene-corrected control cell line, the isogenic control of an SOD1 mutation. For this, we see regular and burst activity. The SOD1 mutation cell line itself shows a less synchronized but more frequent spiking activity. And if we look at the activity of the C9ORF72 cell line, we see a completely different activity that is much more intense, with longer and stronger bursts. When we compare the spike train parameters of our different cell lines, we get the following image, which you can see in this diagram. Here, you can see spike train parameters for our control groups, the isogenic corrected cell line, and a different motor neuron cell line from a healthy donor. In red, you can see the activity parameters from the SOD1 mutation and the C9ORF72 mutation in blue. In detail, we see a lower spike rate for the healthy cell lines than for the diseased ones. We see parameters of the burst structure like burst period, burst area, and the max spike rate in bursts, which separate both groups. We see less regularity of the oscillation parameters for the diseased cell lines and also less synchronicity. Altogether, the SOD1 cell line with a D9OA mutation and the C9ORF72 mutation cell line show a more excited activity pattern than the wild-type cell lines, which is in line with the clinical experience and in vivo models of ALS. How can we now use this for the testing of potential ALS compounds? To this end, we apply a compound after 8 and 11 days in vitro with the medium change. We record the final activity after 14 days in vitro. Here, we have the SID1 mutation in red, its isogenic control in gray, and the spike train parameters for the group treated with riluzole of a concentration of 1 micromolar in green. We see an apparent rescue with riluzole from the diseased group towards the healthy group in all of the presented parameters. To make this better comparable, we project our spike train parameters to a one-dimensional space with the so-called effect score. Here, we have normalized the value of the healthy group to 1 and the diseased group to 0. Now, we can immediately see the potential therapeutic effect of a compound. For 1 micromolar riluzole, we see in our assay a significant partial therapeutic effect. We also have validated these assays with other biomolecular readouts not presented here that support the constructed validation. Furthermore, we have a phase validation. This means we have translated a functional phenotype of hyperexcitation known from the clinical and in vivo experiments into an in vitro assay. And finally, a predictive validation is given by the testing of compounds. The advantage of our assay is that we not only measured an acute effect. In addition, we have applied the compound some days before the final recording, which better mimics a therapeutic situation. Thank you very much for your attention. Please contact us at Neuroproof for further information via email or through our contact sheet on our homepage. Goodbye.